Okay. All right. So Emily Grau from the University of Connecticut is going to tell us about TreeGenes and CartograPlant.
Can you guys hear me okay with the mask on? Okay. So I'm going to talk to you today about TreeGenes and CartograPlant. TreeGenes is a web database for forest trees, and normally at this point I go into a spiel about why trees are important, but I'll skip that. So we bring together publicly available data for forest trees. On top of that, we layer curation by human or many humans. And then on top of that, we have tools for analysis for our users that they can use from start to completion right on their website. Where does this data come from? A lot of our data comes from other primary repositories like NCBI. We also bring in data from Dryad, and we have direct user submissions. So the user submissions that we're bringing in, these are things that are not being held by places like NCBI, or maybe not standardized like you might find at Dryad. So I said it's for forestry genetics. So we are holding really anything related to genomes, genes, markers, etc., but also phenotypes, environmental data, and some related data like literature or things for the community like conferences. And what I'm going to do is I'm going to walk through a little bit about how to use the website and then show some of our analysis tools that we've been working on. So the first thing you might want to do is a standard search. It's up there in the corner, it might be hard to see. That's Pinus. You get results, you can click on them, but there are other ways to access the data. So we have a species overview page. This is the sequence summary by order. We have 16 different orders, 250 families, and over 2,000 species in the database. And here you can see summaries of numbers. So we have genomes, transcripts, we have genes and proteins, of course, and we have markers and phenotypes and our unit genes. You can also drill down to the individual species. And from there, you can click out and look at a species profile. We also have a species page where you can go look at genus and search. And this is what you would be brought to. So this is the species overview page for sugar pine. You can see the tabs up there that allow you to click through to the data that we hold. Sugar pine, of course, has a genome. Most of our species do not. We have, as I said, over 2,000 species, about 180 genomes, which is amazing, but most of these don't have annotations. So most of our species are not going to have a genome tab, for example. We're just going to show you what we have available. But for sugar pine, we'll look at a genome tab. If you click there, right away, you can see the files that are available for download. There's something you can click out that shows you the details about this genome. And what I've highlighted with the circle is a link that you can click that brings us to one of our tools, JBrowse. Ta-da! I'm going to go back, and we're going to click on this other tool. This is our sequence similarity search. It uses Diamond to make things a little bit faster, in addition to traditional BLAST. And this is one of our analysis tools. So from our website, you can provide your sequence, what you want to search against, and all of that analysis gets done on our end and comes back to you. I'm going to use that as a jumping off point to talk about our user accounts. So I know everyone, on the internet wants you to sign up for a user account. I had to sign up for one to pay for parking at a national wildlife refuge. And it asked me for my birthday, which we do not do. Our user accounts are easy and quick. And it allows you to use our tools and come back. There's the analysis section, and you can view the analyses that you have run and get your results. I'm going to come back to our user page. This is also one of the places you can start a data submission to us. So we've worked on a pipeline to allow users to hopefully easily submit data to us. We worked really hard to make it easy, but also to make sure that we're collecting enough information so that it's useful for other people. called it's called TIPS. And to that end, we have used MIAPI, the minimum information about plant phenotyping experiments, to standardize how our phenotypes are brought in. MIAPI utilizes ontology terms, just standard terms that can be shared across experiments so that we can connect data from different experiments. And we have similar steps for our genotype and environmental information. Once your submission is complete, we look at it and approve it, and it goes onto our data submission page. If you click on one of those, it brings you to the overview, where you have a lovely map, and there's an accession, so you can use this for your data accessibility statements and papers. And it's a place where this data will live forever and people can access it, and hopefully it will be useful to someone else. From this overview page, you can click through, see what's available, phenotype or genotype information about the paper, the species. And go back to the species overview. This is the population genetics tab. And this is where our TIP studies are displayed by species. And you can click through here to our big analysis tool that we've been working on a lot, CartograPlant. So CartograPlant is a web-based resource that brings together genotype, phenotype, and environmental data, and allows you to do landscape and association analysis, all again from our computational resources. The data for CartograPlant comes from what we just talked about, but we also bring in data from BN and from TreeSnap. And we have brought in over 900 different environmental layers. So I'm going to give you a quick visual tour of using CartograPlant. Hopefully you can see there's a map with the dots representing our trees. Here's a slightly closer view. We have an environmental layer open, and there's a tab telling you about the layer. Here's another view. This one includes whirlwind and information box has popped up based on where it was clicked. So here I selected a tree, and from here you can view the study that it came from. It tells you what type of data is available for this study. I made a collection of trees here. And I have clicked through to the analysis section. I should note that this is still under development, but it's super duper close. And we're going to be doing a workshop about how to use CartograPlant in a week and a half. So from here, you can filter. You can filter by trait, by phenotype. You can filter by genetic marker. There's a few more that I'm going to skip because I'm not close to the end. And you can select what data you want from the environmental layers to be brought into your analysis. And then you can select the workflow that you want. These have been curated, pre-designed by us to fit the forestry community research needs. And it'll be selected. The available list depends on what data you have already selected, what markers are available, etc. You click submit, it runs, it comes back to you on your user account, and hopefully something good happens. Okay, thank you.